Hello, everyone, and thank you for joining us today for a special event focused on the use of messenger RNA molecule in the development of vaccines and novel therapies hosted by Elsevier. I am Kumsal Bayezid, the CEO of Elsevier, and it is my pleasure to introduce our esteemed guest today. Professor Dr. Uğur Şahin, CEO and co-founder of BioNTech. As a physician, immunologist, and leader in the development of novel approaches to fight cancer and infectious diseases, he is also one of the world's foremost experts on messenger RNA medicines. Professor Dr. Özlem Türeci, Chief Medical Officer and co-founder of BioNTech, who's an immunologist and cancer researcher with extensive translational and clinical experience. Professor Ivan Đikić, Professor of Biochemistry at Goethe University in Frankfurt, and a fellow of Max Planck Institute of Biophysics. And Dr. Richard Horton, editor-in-chief of the Lancet, who is one of Elsevier's treasures and has been leading the Lancet for over 25 years in its mission to be a change agent in medicine and advocate for public health. Uğur will kick us off with a keynote presentation on the scientific and technological hurdles that had to be overcome for the successful application of mRNA vaccines. We will then move to a panel discussion moderated by Richard. The panel will discuss the exciting explosion of mRNA-based drug development currently in preclinical and clinical phase and share their perspectives on the therapeutic use of mRNA for the treatment of cancer and autoimmune diseases. The story of what brings us here today is one of problem solving and innovation through determination, urgency, and collaboration to address a global health emergency. Seeing the first paper published on the novel coronavirus by the Lancet on 24th of January, 2020, Uğur and Özlem initiated Project Lightspeed to develop a vaccine. They then partnered with Pfizer in March last year to produce the first COVID-19 vaccine to be approved for emergency use by December. The vaccine was an incredible scientific accomplishment, going from lab to patient's arms in just 11 months, instead of the 5 to 10 years it can typically take. Their work has given millions around the world hope and faith that we can survive this pandemic. It has also shown what can be achieved when we are all united around a common purpose. I thank all of our distinguished speakers for being with us today, for their significant contributions to the fight against the pandemic, and will now hand it over to Dr. Uğur Şahin to begin his lecture. Thank you very much. Uğur, over to you.
Dear Kumsal, thank you so much for the invitation. Thanks for your team for organizing this wonderful event. And thanks for the kind intro. I'm going to share my screen and I hope that you can see my screen. So I would like to provide a presentation about mRNA vaccines in general, and specifically introduce our work, how we solved in our early research some of the key challenges relevant for the development of clinically active mRNA vaccines. And I would also like to provide you a background at the end of this presentation, what kind of other applications are going on. So I tried to. So the messenger RNA was discovered as in its function 1961. So in 1957, Francis Crick formulated the central dogma of molecular biology. That means the information flow from DNA to RNA to protein. DNA makes RNA and RNA makes protein. At that time, it was clear that the genetic information which is in the nucleus and the protein translation which happens in the cytoplasm needs an intermediate molecule. But the favored hypothesis at that time point was that the RNA responsible for this translation is a stable molecule. And actually the idea was that all the ribosomes were specialized factories, each ribosome making one protein. This changed with the publication and discovery of the messenger RNA, showing actually that messenger RNA is an unstable molecule transporting based on transcription of a copy from the nucleus, and this transcript mediates the translation of the protein, and after mediating the translation, it is degraded. So it was an unstable intermediate carrier of genetic information, and it was proposed to be named messenger RNA. The first 30 years of mRNA research came up with a mix of insights and information and progress. It was about the insights about how mRNA is built, how mRNA is interacting with the inside of the cell. It were discoveries based that mRNA has a cap, and it was also discoveries resulting in the identification, for example, of the T7 polymerase and sp6 polymerases, and production of this enzymes, allowing for the first time to prepare mRNA by in vitro transcription, whereas the early research focused on isolation of messenger RNA from cells and using that, for example, for transfection. I will just jump 3 decades later and will explain you the general concept of messenger RNA vaccines. And so messenger RNA vaccines are produced from a DNA template. And for the DNA template, we use a plasmid, which is linearized and which has a polymerase binding site, for example, T7 polymerase. And this is done in vitro. It's called in vitro transcription. And this creates a messenger RNA, a transcript which has many similarities to the original mRNA in our bodies. Means it has a cap, the 5' UTR, the 3' UTR, and a poly-A tail. mRNA outside of cells is rapidly degraded. To increase the transfection of cells, we use lipids or polymers and form nanoparticles. And these nanoparticles have 2 functions. On the one side, they help to avoid degradation of the mRNA by extracellular RNases. And the second function is they increase the uptake of the mRNA and enable the release of the mRNA into the cytoplasm, where the mRNA is translated. And the mRNA which comes from outside, of course, competes with all other mRNAs in the body. So that means to get to be efficiently translated into a protein, we require a number of modifications to allow that the mRNA which is delivered is indeed translated to sufficient amount of proteins. For vaccines, it is relevant that the protein reaches the surface. It could be secreted or stay at the membrane. And the second aspect, what is important, is that the protein is processed and presented on MHC1 and MHC2 molecules, resulting in the stimulation of CD8 and CD4'T cells. So in the last 30 years, there was a convergent evolution of independent improvement, which allowed to increase the potency of the mRNA. What do I mean with potency? I mean with potency that if we deliver a messenger RNA into the body, we want to get as much as possible protein translated. So, and this can be achieved by a number of modifications, modifying the 5' cap, modifying the sequence around the 5' cap, modifying the untranslated region before the start codon, modifying the 3' region, modifying the poly-A tail, modifying the sequence that is going to be translated, and modifying the chemistry of the cap, or modifying the chemistry of the in vitro transcribed mRNA. And with this type of modification, it is possible to increase, if this is the translation of the original mRNA, modifications could allow to increase the translation, or they could allow to increase the half-life of the mRNA, resulting in the same amount of translation but prolonged, or it could be combined effect. And by combining this type of improvements, it is possible to dramatically increase the translation and the stability of the mRNA inside of the cell. So when we started our research more than 20 years ago, we are cancer immunologists. We were interested to use mRNA for cancer immunotherapy, means to use mRNA as therapeutic cancer vaccines. And one of the important insights at that time was that dendritic cells are the key stimulators of immune responses. So one of the goals were to deliver mRNA into dendritic cells, and this was done and described by other groups in the mid of the 90s. But we were interested in the elegance of the mRNA. Instead of transfecting cells outside of the body and then doing a cell therapy, we were really interested to deliver the mRNA directly for in vivo application, and thereby reducing the steps to just in vitro transcription, purification, and then injection of the mRNA. We hypothesized that the delivery of the mRNA to the lymphoid tissues like lymph nodes, spleen, could allow a particularly improved immune response. And of course, this was a hypothesis, and we started to test it. One of the first series of studies that we did is to understand what happens if we transfect dendritic cells in vitro with mRNA. And what we observed is that we have translation for a certain time, and then the mRNA translation goes down relatively quickly, and we have relatively low translation. So we started to systematically analyze what are the influence of the structural components, the 5' cap, the UTR, the 3' UTR, and we pioneered the systematic optimization of mRNA cassettes by identifying multiple ways of stabilizing messenger RNA, particularly in dendritic cells, because stability of the mRNA is cell dependent. This was a focus of a research in the beginning, but we continued to do that. And in the meantime, by using really discovery approaches, library approaches, we were able to further improve the translation of the mRNA. And this will become relevant because at the end of the day, for making a potent vaccine, it is important to have as little as possible mRNA and induce a strong immune response. The second topic of second focus of our research is the question how to deliver mRNA selectively in dendritic cells, but this time in vivo into dendritic cells in the lymph node and in the spleen. So the first series of optimization resulted were very successful. So here's the approach. Here's again the messenger RNA vector cassette. So this is the antigen to be encoded. Yeah, and these are the structural elements, the 5' cap, the UTRs, and other regulatory elements. And what we did is by systematic testing, for example, we identified modified cap analogs, which increased the stability and inhibited the 5' degradation of the mRNA. or we identified regulatory sequences in the 3' end, which enabled independent improvement of half-life, or for further improvement of the antigen presentation into class 1 and class 2 epitopes, we use signaling retargeting sequences. And this independent improvements, when combined, had a factorial improvement. So we were able to see a several thousand-fold improvement of the immunogenicity in using CD8 and CD4'T cells. So with this type of vector backbones, we went into in vivo experiments and were really surprised to see what happens if we deliver mRNA into the lymph nodes. And this was an experiment by injecting mRNA, encoding for an antigen recognized by adaptively transferred T cells, and the T cells can be stained. Yeah, so this is the stain of the T cells with by immunohistochemistry, and this is the lymph node after injection of the control mRNA, and this is the lymph node in mice which received the mRNA encoding the respective antigen. So this is an antigen-specific dramatic expansion of antigen-specific CD8 and CD4'T cells. So we were able to optimize these cassettes, and at a certain time point, we believe that it is possible to go into applications, into clinical applications. And I will share you a few applications which we pioneered in the last years. And the applications all have the same type of approach. They are based on delivery of antigens. These antigens are different. For example, antigens encoded by mutations and antigens encoded by shared overexpressed antigens, or antigens which are used for stimulation of CAR T cells, and of course in the infectious disease field also viral antigens. We are using currently also mRNA to deliver autoantigens, and instead of stimulating an Th1 immune response, in this case we deliver the mRNA and the lipid nanoparticle in a way that it does not induce a Th1 immune response, but induce the expansion of regulatory T cells. The versatility of the mRNA allows us to do this application in an extremely tailored and precision fashion. Let me start with the development of vaccines against non-mutated self-antigens. And the challenge for cancer vaccines encoding self-antigens is that due to the tolerance, it is extremely difficult to get a strong T cell response upon vaccination. And we believe that we can overcome this if we are able to deliver the mRNA to the lymphoid dendritic cells. Since we saw the limitation of delivery of the mRNA by direct injection into the lymph node, we started to systematically analyze whether we could develop formulations for intravenous delivery, which are based on nanoparticles. So we started a discovery approach and systematically evaluated different lipids. We evaluated different packaging and different concentrations of mRNA and lipids. That means we changed the charge ratio, and we tested different sizes of the nanoparticles, and we discovered that by delivering nanoparticles in the range of 200 to 400 nanometers, which are near neutral, that means that they have the same amount of positive and negative charges, we could enrich the translation of the reporter mRNA in the spleen. And within the spleen, we could see that the mRNA is really translated selectively in dendritic cells, because when we depleted the dendritic cells in an animal model, yeah, then we could see that the signal is lost. So this type of vaccine allowed to deliver an antigen coding antigen directly, systematically into the spleen, but also into the lymph nodes. So we used for this approach an mRNA which is based on uridine chemistry, and uridine mRNA is able to stimulate TLR7 and TLR3, so the pattern recognition receptors, and this stimulation results in induction of interferon alpha. And this interferon alpha is a wonderful maturation and adjuvant signal to stimulate particularly strong T cell responses. And interferon alpha is also well known to inhibit regulatory T cells. So in this model, we were able to overcome the tolerance in the murine model, to overcome the tolerance against self-antigens, for example, tyrosinase, and could show that established melanomas, metastatic melanomas in mice, could be cured, and that was associated with strong T cell responses. So we translated this approach into the clinical setting. And in the clinical setting, we not only use one messenger RNA, but we use a number of 4 different mRNAs encoding 2 well-known tumor antigens like New York ESO1, tyrosinase, MHA3, and one that we had discovered and which were expressed in metastatic melanoma. And these 3 tumor antigens could serve as a benchmarking for immune responses, because they had been tested in many vaccine studies using different formats of vaccines, including of mRNA delivery, for example, using mRNA delivery into the skin. So when we intravenously applied this 4 antigens, and this is one of the key features of mRNA vaccines, you can deliver really multiple antigens. So in this case, we co-delivered 4 tumor antigens in parallel. And what we observed is that the distribution of the mRNA and the lipid nanoparticles in mice, which we have seen to be enriched in the spleen, were also found to be true in cancer patients. So after a few hours, we could see strong activation of metabolic activity in the spleen. We observed the same type of dose-dependent cytokine response. So this is the interferon alpha, the key pharmacodynamic cytokine. And this is a dose weekly dose escalation in a melanoma patient who received in the first week 7 microgram and then increasing dose. And you can wonderfully see the increase of the cytokine levels. And after repeated vaccination, we evaluated the T cell responses that we got. And we observed that despite this type of vaccination, we get much higher immune responses against specific epitopes as compared to immune responses that were described in the past for cancer vaccines. And this included the induction of de novo immune responses, as well as the amplification of existing weak immune responses. We observed for the first time in our clinical trials that this type of vaccine not only induces extremely strong immune responses, but also is associated with single vaccine-based clinical responses, objective responses using vaccine alone, or objective responses in patients who had progressed under anti-PD-1 treatment in combination with vaccination. So this is a patient, for example, who were progressing under treatment with an anti-PD-1, and after initiation of the vaccination, the tumor shrinked. So these are the clinical data. We observed that in patients who were checkpoint inhibitor experienced metastatic melanoma, we had more than 35% of objective responses, which is a pretty high high response rate. The response rate in CPI experienced melanoma is in the range of 10 to 15%. And this was in line with the induction of T cell responses. So this you can see that the immune response after vaccination starts to build up and grows over time. And the T cells that were generated had a PD-1 positive phenotype. So that means it is that this provides a new way in thinking about cancer immunotherapy, providing a PD-1 positive antigen-specific T cell population, which then can be combined with anti-PD-1 treatment in the form of resensitization. The second type of innovation that I would like to show you is a different type of vaccination. Here, the aim is not to stimulate classical T cells, which recognize the antigen by their T cell receptors, but the idea here is to combine vaccination with CAR T cell therapy. Let me provide you the background why we believe that this is important. So one of the key, or let me start with the positive aspect. So CAR T cell therapy is extremely successful in the setting of hematological B cell malignancies. But with regard to epithelial tumors, the success rates are very low. Actually, response rates are in the range of 10%. And one reason for that is that CAR T cells, after infusion into patients, drop, the number of CAR T cells drop over time, and there is no persistent T cell response. So our idea was to combine CAR T cell therapy with a vaccine which stimulates the CAR T cells. That means a vaccine, which an mRNA vaccine, which delivers the mRNA to dendritic cells and enables cell surface expression of the antigen, which is recognized by the CAR T cell. So the idea was now to combine two worlds, the cancer vaccine world and the CAR T cell world. And with that, we wanted to accomplish that we, on the one side, reduce the dose for CAR T cells to be delivered, and then by repeated vaccination, keep the CAR T cells in an optimal therapeutic window and counteract the lack of engraftment of CAR T cells in solid tumors. So we have so far preclinical data for this approach. So in the preclinical testing, we evaluated CAR T cell transfer in mice, and this happens if CAR T cells are transferred to mice, the numbers remain low and drop over time. But if it's combined with the vaccine, with the mRNA vaccine delivering the CAR T cell antigen, we can 100-fold increase the number of CAR T cells. And if we continue vaccination, we can keep the numbers high. If we stop vaccination, the numbers drop. And if we then re-vaccinate, then we can again increase the numbers. The interesting aspect is that we generate here CAR T cells with a typical effector memory and central memory phenotype, and we were able to show that this type of combined CAR T cell and vaccine treatment resulted in rejection of really large tumors by CAR T cell treatment. So this study in the preclinical setting was done with a new CAR T cell target, or with CAR T cells targeting a new oncofetal protein. This is Claudin-6, and Claudin-6 is not expressed in adult tissue, but is expressed in fetal tissue. But it is upregulated in many tumors, like, for example, testicular cancer, ovarian cancer, endometrial cancer, and many other tumors with a lower frequency. So we have now an ongoing clinical trial in Germany and in Netherlands, and are in the dose escalation phase. And this is a basket clinical trial with multiple indications. And we just reached dose level 2 and started with the first mRNA vaccinations. So the last example that I would like to give you from oncology are personalized neoantigen vaccines. And the personalized neoantigen vaccines, a short introduction about the background. It is well known for almost 3 decades that tumor mutations can be recognized by the immune system, either by CD8 T cells or by CD4 T cells. So they are recognized if the mutated epitope is presented on the HLA alleles. And this was an observation where the tumor immunology community did not know for many years how relevant this is for cancer immunotherapy. With the emergence of checkpoint inhibitor therapy, it emerged, and that many patients responding to checkpoint inhibitor therapy have T cells which recognize mutations. So that means checkpoint inhibitor therapy drives anti-tumoral immune activity of neoantigen-specific T cells. Seeing that, of course, the idea is, can we get stronger T cell responses against these mutations? What was published independently by different groups is that only 1 to 2% of mutations are recognized spontaneously, resulting in a situation that only tumors with a high mutational load respond very well to checkpoint inhibitor therapy. Our idea was to ask the question, can we increase the immunogenicity rate by encoding mutations using mRNA? The challenge here is that every patient comes with a different type of mutation set. So this is a simple slide showing how tumors occur. Tumors occur by random mutations, and due to the randomness of mutations and due to the accumulation of mutations, random accumulation of mutations, we have a situation which are complicated in 2 ways. One complication is that every patient has a different type of mutation profile. So if you compare, for example, the mutation profile of 2 patients with lung cancer, the overlap is less than 5%. And the second challenge is that if you look closer, there's a tumor heterogeneity. That means tumor cells have really multiple different mutations, even within a single patient. So the way how precision oncology is executed in our times is to ask the question, if the patient has a given mutation, and if the patient has a given mutation, and then if a drug is available, then this patient might receive this treatment, which is tailored for patients with the specific mutations. We were interested in a different way of cancer immunotherapy, the individualized treatment. The idea that we had is, why don't we really use the broad spectrum of individual mutations? That means come up with a vaccine which really encodes multiple mutations, which are all individual. This approach theoretically has a key advantage. We could go for much larger target space, antigen space, and the second advantage is we can tailor it really to the individual patients. So we evaluated this approach with first publications in 2012 and follow-up publications in 2015 in animal models. And what we worked out is a generic approach for personalization of cancer vaccines. It is based on sequencing the tumor, identification of the tumor antigens, of the mutations, then using computer prioritization to identify those antigens which are expected to be presented on MHC class 1, HLA class 1, and HLA class 2, and select the top 10 or top 20 antigens, mutations, and use these mutations to form a poly-epitopic vaccine, which can be delivered to induce immune responses against multiple mutations. We have shown that this approach in the preclinical setting in different tumor types is profoundly active, inducing immune responses in the range of 40 to 60%. And we have particularly observed also strong CD4 T cell responses. So we translated this approach into the clinical setting in melanoma. This was our first clinical trial with the first patient recruited in 2014. And it's the same approach as in the preclinical setting. We get the tumor sample from the patient, we get blood, use both to do sequencing, identify the mutations, use the RNA sequencing analysis to identify which mutations are translated, and then use computerized computer prediction tools to prioritize the mutations, make the tailored DNA template for the individual patient, and then produce the mRNA. And these steps, since this is really in vitro transcription, can be done in a few days. So we optimize the process, and since the patient has, of course, a growing tumor, we needed to implement an extremely fast process. So we, in this first trial, we treated 13 patients, observed that we got immune responses against 60% of mutations, CD4 and CD8 immune responses. We observed particularly strong T cell responses. These are T cell responses just ex vivo, that means not amplified in vitro, directly in the blood of patients, which means that billions of T cells were generated by this type of vaccination. And as a clinical activity, we observed in the treated patients that the metastatic relapse rate, and these patients had multiple metastatic relapses before entering the vaccination, the metastatic relapse rate dramatically dropped. And by comparing the metastatic relapses before vaccination and after vaccination, we saw that this is significantly different. So this was 2017, and since then, we further improved our technology. So we reduced the manufacturing time from 3 to 5 months to 3 to 5 weeks to deliver a vaccine, starting from the sequence. And this approach is now being evaluated in randomized clinical trials in melanoma as well as in colorectal cancer. So this was our cancer work, and in 2020, when we started 2020, of course, our focus was to continue our cancer work. But then this new outbreak happened in Wuhan. I heard from this outbreak already mid-January, but I was not concerned because this type of outbreaks happen every year, and usually they are controlled, and publications indicated that the measures were able to control the outbreak in Wuhan relatively quickly. Yeah. And one of these measures were really exit screening for people who wants to travel, so that everyone, for example, who had fever were not allowed to travel. And so I was, of course, interested, but not concerned. And this changed when reading online paper which came out in Lancet. Kumsal referred to that, and this was a Friday evening, and it was the first publication that I saw which described these infections in a familiar cluster. A family who traveled to Wuhan and traveled back. They were infected and they got the infection in Wuhan, and they came back and infected other family members. And this was an excellent publication showing the human-to-human transmission, showing that this is a new SARS coronavirus, showing that the PCR testing works. And with the additional information, it was clear that the outbreak is running more than 6 weeks in this Chinese megacity. But what was really concerning is that one of the infected patients were PCR positive, strongly PCR positive, but asymptomatic. Yeah, and the conclusions from this paper were that this virus, this new outbreak, fulfills the full pattern of a pandemic outbreak. It is the whole human population is expected to be susceptible, even with the conservative assumption of a relatively low reproductive number and relatively low significant mortality that I had calculated. It was clear that this could become a global pandemic because of the aspect of virus spread by asymptomatic individuals. Just to remind you, the measures that were one of the measures to restrict the infection was keeping patients with fever in Wuhan, but this was not relevant for the asymptomatic travelers. So I was extremely concerned, did some research, understood that Wuhan is a mega city, understood that this city is well connected to the rest of China by trains and by an international airport, yeah, and saw the very high likelihood that this outbreak is already spread almost worldwide. And on the same weekend, we started to do a plan to develop a vaccine, because what we knew was that we have a technology in our hands which allows extremely fast tailoring of a mRNA vaccine, because it was optimized for development of cancer vaccine in the shortest possible time. And we had experience in stimulating strong immune responses. So we started to evaluate what we can do. And we are not virologists, so we dived into the literature on this weekend, read almost 100 papers, yeah, came up with an understanding that the prior coronavirus family had targets, the spike proteins, which could be suitable for vaccine development, learned that there are different forms of spike vaccines, the receptor binding domain or the full spike, but unfortunately there were no clinical results available. So that means we had to design the vaccine. It was not clear whether the full spike or the receptor binding domain is fine. And at that time, there was also a great concern that vaccines against coronaviruses could even do more harm than they are useful. So this antibody-dependent enhancement or antibody-dependent disease worsening was one of the concerns published independently by different groups. So the only way to deal with this uncertainty was that we came up with 20 candidates. So we used our cancer vaccine platform to set up a parallel testing approach. We started the project, named the project Lightspeed, asked our teams to set up a 24/7 program with working shifts in the evening and also on the weekend, and reduced all lag times, and enabled within a few weeks to reduce the number of candidates to 4 candidates, which showed strong preclinical activity for clinical testing. And shortly to explain you the mode of action in a simple manner, how such a vaccine works, an infectious disease vaccine. This is unfortunately missing in many textbooks. So the vaccine is delivered as a lipid nanoparticle, where the mRNA is encoded. mRNA is encoding the protein, and interestingly, many people believe that the muscle, if it's delivered intramuscularly, is the main organ for vaccine activity, the translation. This is not true. It is. We have optimized our mRNA delivery to deliver even after IM injection into lymph nodes and deliver to dendritic cells. And we have again this aspect of inducing CD4 helper T cells and CD8 T cells by different presentation pathways, and by secretion or by surface expression of the antigen, induction of virus neutralizing antibodies. It is well known that virus neutralizing antibodies decline over time, whereas, for example, if the vaccine is properly engineered, T cell responses could stay really very long. And this was the aim, to really induce a vaccine which is able to induce CD8, CD4 T cell responses as well as antibodies. This is just showing you what happens if you deliver such a lipid nanoparticle vaccine into the muscle in mice. This is the RNA in situ analysis in the muscle. Is a relatively little uptake, but the draining lymph node is full of antigen, yeah, and in the draining lymph node you can see that the antigen is also enriched in follicular dendritic cells and also in conventional dendritic cells. This is just the immunohistology showing you the staining of the draining lymph node as well as the muscle. So we have here again the same principle. So we went into after having successful preclinical studies, we started a clinical trial in Germany, and we entered into a partnership with Pfizer because we knew that our organization is not suitable to do this large phase 3 clinical trials, and that and also that a vaccine needs to be distributed. So our two goals were to develop a vaccine in the speed of light in the shortest possible time, and to enable to scale up manufacturing to enable worldwide delivery of our vaccine. And this is the selected candidate. It encodes for the full spike protein with 2 mutations described by Barney Graham, which expose the spike part better to induce stronger antibody responses. We have multiple elements that we had discovered in the past. And the vaccine was based on a nucleoside modified mRNA. This is a modification which dampens the immune response and increases the tolerability introduced by Katie Carrico and Drew Weissman. So the vaccine really combined the best of different worlds. And we were able to see that even lowest doses of the vaccine, even 1 microgram, results in a prime boost, results in antibody responses. And we have seen in the younger population that 10 microgram and 30 microgram and 20 microgram of vaccine has relatively equal amounts of neutralizing antibodies. But in the elderly patients, we realized that we need to go for 30 microgram. And what is really important is that we also observe T cell responses and the importance of T cell responses. We are all discussing about variants, and these variants, of course, are in part antibody escape variants, but it is extremely difficult for a virus to escape T cell response because the T cell responses are based on HLA polymorphism, and every individual has different HLAs. Yeah, so therefore we analyze whether we get this polymorphic T cell responses, which is the case. So this is an individual vaccinated with BNT1 and 62b2, and these are the different domains, peptides for the different domains. And as you can see, no T cell response before and strong T cell response after vaccination. And these are controls, like for example, T cell responses against cytomegalovirus, EBV, and flu. So that means we get really a very strong T cell response. And this is just another depiction. This is a method which is called multimer staining. And here you can again see these are different individuals. This individual has an HLA-2402. This one has a 3501, and this is a HLA-02 patient. And you can see different sequence parts of the spike protein are presented and act as immunogens here. And therefore, there is no general way to escape a virus even if it's a variant virus, from T-cell responses. And T-cells, and that's what we believe, are the protective shield against severe disease, whereas antibodies are the protective shield against infections. So most of you know the clinical data. As we were, of course, when we saw for the first time saw the data, were extremely excited. We expected efficacy in the range of 70%, and we were also prepared that the vaccine is going to fail because of the biology, unknown biology of the virus, but the biology was in favor of a good vaccine-mediated control, and we saw this extraordinary efficacy in the phase 3 clinical trial. And of course, with this efficacy, having an effective vaccine meant that we have to manufacture vaccines. Luckily, we had already built a scale-up technology for our cancer vaccines, and the scale-up technology was based on just producing about 10 to 20 grams of mRNA. But luckily, the technology proved to be stable for mRNA production, so that we could use larger fermenters and could just adapt the protocols slightly to allow to get much higher doses. So 2019, this was the amount of doses that we produced in 2019. In 2020, it was 25 million doses. And now having the complete network of Pfizer, BioNTech, and many collaborators, we will be able to deliver more than 3 billion doses. And this is based really on the limits that we can produce about 90 kilograms of mRNA. And the low dose, the 30 microgram dose of this vaccine, and this is really one important aspect of the optimization. The low dose allows us to deliver this huge amount of vaccine doses in 2021, and in 2022, we believe it could be more. So let's talk about variants. At the beginning, we started to evaluate variants with single mutations and could see that single mutations do not have any impact on the neutralization titers. So this is an in vitro assay where we use sera from vaccinated subjects and test whether the sera could inactivate and inhibit the uptake either of real viruses or pseudoviruses. So this was the first analysis of a complex variant, the alpha variant, or B.1.1.7, showing that this is also a stable response. And in the meantime, also together with our partners Pfizer, we continue to analyze all emerging variants of interest or variants of concerns. So we have the alpha, beta, gamma, delta, eta variant. We are evaluating all of them. And of course, what is at the moment the most interesting variant is the delta variant, the B.1.617.2. As you can see, as compared to the wild type, we have a reduction of the titers. It's about 50 to 60% of this titers. So the vaccine is still active, but with a lower neutralization titer. And this is one way to evaluate potential vaccine efficacy in the laboratory setting. And the other way, of course, which is more important, are real-world studies. This is a study evaluating vaccine effectiveness in Qatar, where our vaccine was used. And in this study, the most important finding was that our vaccine is still fully effective in preventing severe disease against the South African variant, the Beta variant, and highly effective in preventing infection. This was a study which were published online 2 weeks ago. It's the real-world effectiveness study in UK, showing that early after the second dose, subjects early after the second dose have a high effectiveness of 88%, also against the Delta variant, and of course full effectiveness against the Alpha variant. So in summary, this was the fastest vaccine development in the history of medicine. For sure, the speed benefited from the international collaboration, from the versatility of the mRNA, as well as on the processes that we had developed for personalized cancer medicine. It helped us really not only to come up with a single candidate, but to come up with 20 candidates and select the best one, thereby come up with a vaccine candidate which is effective as a very low dose. And we can use this technology again, as shown for the personalized cancer vaccine field, where we can deliver any type of vaccine to any type of sequence within a few weeks. The same technology can be used to adapt the vaccine against variants. So with that, we have now closed the gap in the pharmaceutical field. So protein therapeutics are available since 1982. The first gene therapies were approved in 2016, 2017, and the first mRNA therapy was approved at least conditionally approved in 2020, and many more vaccines and therapies will come. This is from one of our reviews that we published in 2014 about the potential therapeutic applications of mRNA. I showed you several applications for cancer immunotherapy, and now we know that infectious diseases, the first vaccine is against a pandemic virus. We have, in the meantime, we are using antibodies encoding antibodies against infectious disease pathogens, cytokines. So we are in clinical testing with several of these concepts. mRNA vaccines have been used for allergy tolerization. They have been used to deliver proteins, either proteins which are natural proteins to induce, for example, to reduce inflammation, or for example, bioactive hormones, or transcription factors. One application field is to deliver mRNA encoded genome engineering enzymes like transposases or CRISPR-Cas, again benefiting from the transient activity of the mRNA. And one really exciting new field is, of course, the delivery of transcription factors. One of our very early applications was to deliver the Yamanaka factors as mRNA and show that mRNA is suitable for reprogramming of cells. And there is not only one mRNA format available. So in the cancer field, we are benefiting from the strong adjuvant activity of the uridine mRNA. The nucleoside modified mRNA comes with a great promise for all applications where you want to avoid an immune response, or where you want to have a more tolerable vaccines. We are working on self-amplifying mRNAs, where you can deliver little mRNA, and it is after delivery the mRNA starts to amplify, or a safety improved version of a trans-amplified mRNA. And of course, this is just the mRNA field. What I did not mention is the field of mRNA therapeutics benefited hugely from delivery technologies, lipid delivery, polymer delivery, and it is going, of course, to become more and more specific. We will have organ-specific applications of mRNA in very near future. So I would like to thank first organizations which supported our research. So we started in an academic environment with 2 students and 1 postdoc working on this project. We benefited from funding from the German government, from the European Commission, yeah, and from programs allowing us to start a company, and of course we benefited from funding from investors. I would like to acknowledge the scientific team. This is of course not possible to name all of them, but I want to name the most important. Our cancer immunotherapy team is with Mustafa, Sebastian Rauf, yeah, our infectious disease team with Annette, Alexander, our T-cell team with Matthias and Lena. I would like to thank our mentor, Christoph Huber. I would like to thank Katalin Keriko, which provided the nucleoside modified mRNA, and of course I would like to thank Özlem, with whom we are doing this journey now for 30 years. I would like to thank the colleagues from Pfizer, particularly Katrin Janssen, Phil Dormitzer, and Bill Gruber, who is the head of the clinical development. I would like to thank colleagues who supported us with formulation from another biotech, Andreas Wagner and Dietmar Kattinger from Polyimmune. I would like to thank for the discovered lipids that we use for the study, Acuitas, Tom Madden, and Chris Barbarossa. And of course, I would like to thank the agencies, yeah, who with us worked in light speed, making that possible. And last but not least, I would like to thank our clinical investigators and voluntary study participants. Thank you for your attention.
Professor Şahin, that was absolutely fantastic, a tour de force. My name is Richard Horton from the Lancet, and it's a pleasure to have this opportunity to thank you on behalf of over 2,500 people who tuned into your excellent lecture. I've got a whole host of questions that I would like to give you. I wish we were together in a room face to face, because I think you would have a standing ovation. But if I may, on behalf of everybody, I'm going to give you a round of applause, because seriously, we shouldn't forget the common courtesies that we have forgotten over the past 15, 16 months. The way we're going to do the discussion is that we've got 2 people to join us, 2 very special people, one of whom you just mentioned. So if I could ask Professor Özlem Türeci to turn her video on, and Professor Ivan Đikić to turn his video on. Now, Kumsal very kindly introduced them both at the beginning. Let me just briefly recap. Professor Türeci is a physician and scientist, and with you co-founded BioNTech in 2008, I believe. She also has an appointment at the Johannes Gutenberg University in Mainz, and she's led the project Lightspeed that you've discussed with us about developing and distributing the vaccine. Perhaps more in popular media terms, you have both been awarded the Financial Times People of the Year, and a very unusual accolade from our world of science. You made the front cover of Time, which really means you've made it. So congratulations on that. Professor Đikić is a professor of biochemistry at Goethe University in Frankfurt, originally, I think, from Zagreb in Croatia, beautiful, beautiful city. A fellow also of the Max Planck Institute for Biophysics, and his main interest is to study the molecular basis of a wide range of diseases, from cancer to neurodegenerative disease to infection. He has many international affiliations, and he's won the Ernst Jung Prize for Medicine and the Leibniz Prize from the German Research Foundation. Now, the way we're doing this, I'm going to invite Professor Türeci and Professor Đikić, first of all, to give some reflections, partly on what you've just said, Professor Şahin, but also from their own perspectives, their own experience. And after that, we will go into some questions. And if I could encourage the audience to post their questions in the Q&A, then I will try and take the most relevant ones. So I'm going to ask Professor Türeci to go first. It's wonderful to see you.
Thank you very much, Richard, also for this warm welcome. It's a pleasure to be here with you and Ivan and Uğur. Yes, it's difficult to add additional reflections to what Uğur has just summarized. And in fact, I was also a bit breathless, because you can imagine when you are in this journey, you cannot really reflect, and having someone show this flashbacks, only then you start to reflect. What I would like to highlight is that for me personally, this journey was a very important learning exercise, because it showed two things. On the one hand, it showed that science and technology in a dire moment for humankind can make a difference. And what I would wish for the future is that we should not wait for such baptisms by fire, by crisis, for many very promising technologies and scientific insights which we have, to enable them to make this difference. So this is one learning. And the second learning, which was important for me, was this cooperation, the spirit with a common sense of urgency, with a common goal, cooperating across nations, across also public-private borders. Yes, we have seen these private cooperations, all the companies, for example, with which we cooperated, these cooperations made this feasible, for example, with regard to the manufacturing piece of this and supply piece, which is very important to achieve what we were interested in from the very beginning, equality of distribution, but also the public-private partnerships of regulators, of governments, of health agencies working with us as private companies. So these are the learnings, and I hope they will help us in future.
Thank you very much. Let me turn to Professor Đikić, please.
Thank you, Richard. Thanks also for Uğur. This is fantastic talk. And I think I'm just going to reflect that what he says didn't happen by accident, didn't happen by overnight. It happened by 20 years of investment in this new technology, which has matured so tremendously. And I think this is what we need to keep in mind. This type of events, when the health is attacked by an enemy, wherever this virus, bacteria, we need to be prepared. We need to be prepared to react fast, to react cooperatively, and to really engage science and technology together. And I think this has truly happened in this corona crisis, because scientists have reacted tremendously. And I think we did well because we are educated to share, to be open, and to work together. And this is, as Özlem says, this has been beautifully seen by their work, by BioNTech collaborating by academics and also the pharma partners. And the second reflection I would like to put together is that, you know, from this we need to learn to invest now, to work now together, and to really do not forget the basic science coupled to technologies. This is absolutely essential for our future capacity to react on time and fast and efficient.
Thank you very much. I think for most members of the public who have followed this story in the general media, they will not have fully understood the history of the basic science that underpinned the vaccine production. I think the common public narrative has been, we've produced this vaccine in a very short space of time, but there hasn't been the discussion of your, as you described it, 20 years of effort. And I wondered if you could, this is to Professor Şahin and Professor Türeci, if you could just reflect on that 20 years. It can't have all been smooth. I mean, the way you presented it, it was one long continuous development and linear progress. But I suspect that there were moments where there were dark days, where you wondered whether you were on the right track and whether you should change, do something different. And then there were moments when you were genuinely elated, when you knew that you got something that you could take forward. Tell us a little bit about the story of the science, the highs and the lows over that 20-year period.
Do you want to ask them? Or
yes, I can start. Definitely, Richard, this was not a smooth process, and every scientist who is on this call in this virtual meeting knows exactly how our last 20 years have looked like. But every scientist also knows that this is the mode of action of scientists. It's not about crying over failures and being paralyzed by desperation about them. The way we work is that we know that innovation means unknown territory, and that we know that we have just to keep going and produce those ripples in this unknown territory and react on them. So yes, numerous what maybe non-scientists would call failures, but each of them contributed to a better learning and to the improvements, both technically, so all those experiments and high throughput screenings to turn each rock which was connected to RNA and put it back in an optimized way. So the technical failures and learnings and improvements, and then also successes, but also those failures and learnings and successes in creating the right environment to do this sort of translation, to have a technology, but also enable ourselves to bring it to patients, to develop it in a sustainable way according to regulatory frameworks, which meant for us unusual career paths. Yes, it was indeed a journey with ups and downs.
Professor Şahin, do you want to add to that?
Yeah, I fully agree with Özlem. The only thing that I would like to add is at the end of the day, all the things that did not work help also to inform us what is really needed. The beauty is that this mRNA is a natural molecule. That means every time when we find something which works better, there's a biological reason, a mechanism behind that. And this is really nice. It is not dealing with the technology. It's really dealing with biology. And then you are connected, as Ivan said, you are connected to the rest of the world. Even though if you are a lonesome mRNA researcher, you are connected to the rest of the world by this biology.
And Professor Đikić, I'm interested from your point of view, and bring us into the world of your scientific research, the connection between basic research, which you pointed out, and more clinical therapeutic applications. Explain a little bit about those connections and how the public needs to understand those, could understand those better.
Right. So many things happen in life without predicting. It's a lot of serendipity. So I think we worked a lot last years on bacteria, how bacteria produce the pathogenic effectors to take over not only the damage the cells, but also to control the host immune response. So when the crisis came out, we shifted the entire research, largely because the labs were closed, so we could do only coronavirus research. We moved the same concept of thinking to viruses and asked the question, what are the weak spots of these viruses? What shall we do to not only target COVID disease? And at that time it was only that, but now we think, how can we use all of this enormous knowledge that many scientists around the world collected on the transcription, on translation, on the proteolysis of the proteins which are required. So we worked on enzymes which are proteases, which are required for processing of open reading frame polypeptides. And these are essential, called main and papain-like protease. And just beautiful work by really many collaborating labs. Within 2 months, we have solved the structure, explained how this protease acts on the side of the host immune response and on the side of the viral processing. And this actually gave us a lead to say, wow, maybe this type of therapeutics might be beneficial to block both sides. Virus does not replicate. And in the same time, we increase the remaining few viruses which are producing interferon response, for example. So that's where we are going now. And we are now expanding it, trying to look. And it's nice to see again the partnership of BioNTech with Pfizer. Pfizer has already come with the new inhibitors, again, the main protease, which will, at the moment is in the first clinical trial, they promise by the end of the year to be ready to go to the clinical trial 2 and 3. And many people in public, they say, why so fast? This is very suspicious. The first is because you have so many clinical patients and clinical material that you can actually do high number clinical trials that you can trust. This doesn't happen ever in the clinical work, and Uğur can actually confirm that, and Özlem as well. So I think these are the moments where we feel as a scientist being very valuable. And I would also like to say here is by having this common enemy, we made the togetherness to work together between postdoc students from different labs, and they became friends. They are really working together. I think this is a fantastic experience. In the light of crisis, I believe we are now much better and ready for future.
Thank you, lovely. Now, I have many questions about the present and the future, but I do just want to look back briefly, because there are two parts of the story that really fascinate me. The first part is this. Today, immunotherapy has transformed so many aspects of clinical medicine. I mean, it's therapeutics which just wasn't around 5 years ago. It's remarkable. But you've been on this journey of immunotherapeutics for 20 years. What was it 20 or more than 20 years ago that taught you, that triggered this idea that the immune system could be corralled, brought to bear on human disease? Because it wasn't an obvious insight 20, 30 years ago, as it's become so common today. What was the turning point?
So for me, there was no actual turning point. It was an understanding or an hypothesis. Cancer is really a complex disease. And if you want to deal with a complex system, you need another complex system. Yeah, and therefore I did not see a rational way to develop a treatment, a universal treatment, which could deal with this complex disease, because it will always be dynamic. Yeah. And I was just struck by the beauty of our immune system, with this hundreds of different cell types, with this billions of immune cells, with the communication of the immune cells, how the immune system is able within a few days to deal with a virus which is able to amplify within 20 minutes. So this is all this beauty which from the viral immunity field, yeah, convinced me that the immune system should also be able, yeah, to deal somehow with cancer. But we had to understand what are the mechanisms needed, and what are the mechanisms which counteract that.
Very nice. Professor Türeci, did you want to add to that?
Yes, I can only agree with Uğur. It's really the understanding that cancer is so complex that you need a complex counterpart. And as Uğur said, we saw the potency of the immune system as physicians, as physicians who treated patients who had viral infections, also patients after transplantations, which failed, for example. Yes, this showed us how potent the immune system could be, and therefore there was this dream: how can we repurpose it against cancer? And you're right, back then we were a very small community of immunologists, of cancer immunologists, who believed in this science fiction story. But you can see the community became larger and larger, and with the checkpoint inhibitors had their first successes, and will continue to grow.
Yes. The second strand, historical strand, I think is also very relevant to a present challenge today. You talked about the importance of fusing academic research, traditional academic research, with a biotechnology focus. I think before BioNTech, correct me if I'm wrong, you had another company called Ganymede, is that right? Now, why did you both take the jump out of academia into effectively a biotechnology startup? What could that do for you that an academic setting couldn't deliver for you? And I think there's a big learning point here for people worldwide about the synergy between academia and the biotech industry. But again, tell us your story there.
I think we have to disappoint you here, Richard. We never did the jump. We never did this jump. We cultivated a life of being wanderers between these worlds, which are so different but complementary, and have to be married in order to really do sustainable development of medicines, which at the end really end up at the bedside. There is no other way to do this, and we understood this very early on, that just with funding of academic research, we would never be able to fund huge clinical trials, which are needed for good reason, and regulators ask for them. And we understood that also manufacturing would be a topic which needed to be funded, and that one would need a special framework which only a company could give us, namely people experienced in working with regulators, setting up drug development processes, complying with the regulations, which also for good reason are in place. And but we did not want to give up our home in academia, and managed to balance both. And I think that for us was important, and I can imagine for many scientists who want to do this step of doing the translation from science to survival, this would be the best step to do, not to lose any of these worlds.
I love that idea of wandering between communities. It's not a choice either or, it's both, it's all, and you're taking a step at different times in your career in and out of those. Professor Đikić, is that the same in your world, that you're wandering between an academic setting and a more entrepreneurial research setting?
Absolutely. I have been now for the 3 years at the part of the Genentech. I was having an appointment in Genentech for 3 years and still kept my appointment in Frankfurt as a professor. That has been incredibly important training process. There I learned that the diseases should be treated more commonly, mechanistically, rather than isolated as, well, here is an infectious department, here is a cancer department. And the beautiful work from Özlem and Uğur says it all by utilizing mRNA technologies from cancer. They have tremendously helped infectious diseases. And this is what experience is needed for young students, postdocs. They should not be grouped, oh, this is an academic career, that's an industrial career. They should take challenge, the scientific challenge, the question, where can they learn it best? And for me, going to Genentech as a scholar was, I wanted to learn the world behind the place where the technologies are developed and how you can go to clinical trials, which you don't know and you don't see every day in your work. And then you recognize how important is the other side of the work, which you normally take as granted. And you think that everything that you do in your lab is exceptionally important. It's not. It is all of these, the almost like a world of things around you that you need to acquire just by experience or by working together that will make you better scientists.
Thank you. Let me turn to some of the issues that are on people's minds today. And perhaps I could begin with you, Professor Şahin. And I was very intrigued by your statement about the importance of the polymorphic T cell response. One of the big debates today, of course, is the fear of variant escape from vaccine efficacy and the importance of suppressing community transmission to reduce the risk of variants emerging. But perhaps we're wrong about that. Perhaps we should be more relaxed about variants if we believe that that polymorphic response is going to provide durable protection in the long term. What do you think about that?
Yeah, we have two goals as a community. On the one side, really to reduce the number of severe diseases, and T cells and long-lasting T cell responses are expected to be able to deal with that. But on the other side, the decline of the antibodies will create a situation that the community is not immune against infection. And if we have sustained infections, yeah, this will increase the mutation rate. Yeah? So that means the wise approach would be really to ensure that we are not only being relaxed and say, oh, so severe infections are not a problem, but ensure that this virus does not continue to mutate, yeah, and reduce that. And the best way would be booster immunizations, and booster immunizations could be done with the existing wild type, or could be adapted to the new variants. And this is, I think we are going to see that.
No, that's very good. It is true, though, isn't it, that not every mRNA, we talk about mRNA vaccines as if they're a rather homogeneous group, but they're not. Even for COVID-19, the CureVac mRNA vaccine, we could say it's a disappointment or it's a puzzle. What has made your mRNA vaccine successful, do you think, whereas other mRNA vaccines have not been? Is there some fundamental biology that we can understand better as in trying to explain why some vaccines have worked and others haven't?
As you said, Richard, mRNA vaccines are not created equally. And even our mRNA vaccines, we have an entire toolbox, and we have which allows us to use mRNA like a shapeshifter. We can sort of tweak it in every direction, and we selected the right combination of features, obviously, for our vaccine. As Uğur has pointed out, we started with 20 different versions, which differed in all those features. We narrowed them down to 4 and tested those 4 in clinical trials, and also there created the data which led us to the right one. So it's really about having the right combination of chemistry, of backbone elements, of the lipid in order to generate the right pattern. So of immune response, the right phenotype of T cells, the right TH type of CD4 cells, and so on. And therefore, it would even not make sense for us to try to deconstruct what others may have had done wrong with their mRNA vaccine. It's simply not possible to do.
Thank you very much. Any other comments on that from either Professor Đikić or Şahin? The heterogeneity of mRNA vaccines and what we can learn from that.
Yeah, maybe just as a comment, we have the protein therapeutics, and we don't say these are proteins. Yeah, right. So we really should see each individual protein therapeutic as an individual molecule, and we know that some antibodies, even if they are antibodies, fail, and others are highly successful because they are engineered better. The targets are optimized. So we have seen, for example, for SARS-CoV-2 antibodies, that several projects failed, whereas some projects were successful. And you can also ask the question, why is that so? And the answer would be, it's really at the end of the day, the optimization, yeah, and the hard work in the preclinics to ensure to get the best possible molecule for clinical testing.
Thank you. Professor Đikić, did you want to add?
I was just trying to say that this is exactly what science is, more the merrier, and you need to try many different things. We are targeting now few diseases. New diseases are going to bring new challenges, and I think more knowledge we have also on the failures, which Özlem was really nicely talking about history. Even that some like CureVac, you say they fail. Maybe they fail, but they learn something out of it, and they might be successful in the future for the whatever a new indication they will go after with the same therapeutics. But what is important to stress today, mRNA therapeutics have actually come in the center stage with amazing opportunities and investment, and I think this is going to be a revolution in the years to come.
Thank you. I wanted to turn to the issue of access to vaccines and the inequity that the world is currently facing. It has been very perplexing to see that people have not understood that the unit of action here is the world, not a country or even a continent. And you spoke eloquently about the scale up to 3 billion doses for this year, but even 3 billion doses, of course, is short of what the world needs. How do we accelerate access so that everybody in the world has the opportunity to get inoculated? Because right now the prospects are not looking good. Who would like to try that? Let me ask Professor Şahin. Yes.
Yeah, I may start. So we feel really the duty to produce as much as possible doses to deliver vaccines. So our initial plan with Pfizer was 1.3 billion doses for 2021, and we came together with our team and asked the question, how can we increase that? Because some vaccines which were expected to come in with our vaccines were delayed or failed, and this increased the proportion that we had to deliver. And the team came up again with innovation. Yeah, so they were able to add an additional layer of scale up, yeah, to make the production more intense, and we increased the production to 3 billion doses. And in the next 18 months, we plan to deliver 2 billion doses of this to low-income and middle-income countries. And we have delivered already our vaccine to more than 100 nations. So this is really not only delivering Europe or the United States, it's really a worldwide supply. It's very close to our hearts. Yeah, and we consider to further increase the manufacturing, and what we did is we did also partnerships. So we have in the meantime more than 20 partners, yeah, supporting us with steps. Yeah, but this partners support us with supply, support us with fill and finish. Yeah. However, to set up a new mRNA manufacturing somewhere else requires at least 1 year. So we plan to do that in Singapore. Yeah, and Singapore is of course one of the most advanced biotech regions worldwide, and still it will take 2 years to have it up and running. So we need to continue to supply from the existing manufacturing nodes. We hope that the other companies will deliver more and come in. I am confident for 2022, to be honest. Yeah. Okay. I believe 2022 will be much better than 2021. Yeah, and even in 2021, we will be able to do significant supply of these countries.
Well, that's very hopeful. That's very hopeful indeed. Any comment, Professor Türeci?
Yes, Uğur already mentioned the most important facts which should be conveyed here. What I may add is everyone understands that the process from starting our vaccine development to its emergency use or conditional approval was indeed light speed. Because there are benchmarks against which you can compare development of a measles vaccine and so on. What is happening in the manufacturing space, the upscaling of capacities for mRNA vaccines, this is happening the first time in this scale, in this dimension for any technology. So it's difficult to appreciate that. In fact, what is happening, and Uğur just said that we have overperformed, even though we very carefully promised 1.6 billion doses, yeah, we have nearly doubled that, yeah. So also there, rest assured, everyone involved, all our partners, and we have more than 15 or 20 in the meantime who are involved in this upscaling, are doing everything which can be possibly done. I'm also confident.
That's great. No, that's great. It's an unfair question to ask because a year ago we wouldn't have even known we would have had a vaccine, as you say. So now to say, well, we need to vaccinate 7.5 billion people by tomorrow is a very tough question to address. Let me just ask you about looking forward. Are we going to have different vaccines to prevent COVID-19, mRNA vaccines to prevent COVID-19 in the future? Do we tweak the vaccine, go for booster doses? What do you see as the evolution of this technology in the prevention of this pandemic?
So I can start. So the key question is, so we know that we can adapt our vaccine, yeah, and this is possible very fast. But what we observe is if we do a booster vaccine with the wild type, we increase the neutralization against all variants. So a booster vaccine, booster dose might be or is most likely to be sufficient against the existing variants. In the coming years, I hope that the scientific community and the regulatory authorities have a mechanism in place to decide, yeah, which should be the yearly strain, or which should be the yearly strains to be selected for a booster vaccine. Yeah. This is not yet established. So regulators are working on that, and it might come end of this year. And if we have this guidance, we can act in a consistent manner.
So a little bit like seasonal influenza.
That's most likely, yes.
Yeah, okay. Now we've been going an hour and 3/4 almost, so I'm going to come to a final question for the three of you. And it comes from one of our audience, and in many ways it has nothing to do with what we've been talking about. Let me be very direct with you. The three of you have been inspirational in your work, in your careers, and today. And we want to inspire a new generation of scientists. Science is exciting. It's, as you said, science is beautiful. And science can change lives and shape the future of society. So one question that came through is, what's your advice or thoughts or reflections for students in lower middle income countries and developing countries who might hope to be scientists? That's a really tough question to end with, but let's end on an inspirational note for the global scientific community. And maybe I start with Professor Đikić, and then I'll go to Professor Şahin, and then Professor Türeci.
I think the question, Richard, is very important because it comes to this disequilibrium of the disadvantaged countries, while the talent is then divided, and the talent doesn't have a great opportunity in those countries. But what I would like to say today to younger people around the globe, they should not be blocked by anything. They should actually come in contact with the people around the world. They should be motivated to come out with ideas that where they want to work. It can be now from Singapore, US, Canada, Europe, as well as in those countries where they want to stay. There are the nuclei of good science, or scientists who have been trained abroad, came back. There is great opportunity. The only stuff that young people need to carry is they need to have a will, wish, and convincing energy that they will go through the career of science. And then there are no borders. I mean, I am an example of a country that was in the middle of the war. Yugoslavia was torn apart, 1991, 1992. I just finished medical school, and I said, during the war, I stayed there. But as soon as I had an opportunity, I wanted to see a new horizons ahead. Went to the US, from US to Sweden, now I'm in Germany. And this I could never predict when I was 24 or 25. So the message is, don't limit yourself, trust in your talents, just go ahead, but keep contacts with your country. I really feel that this is important. We are international as a scientist, but we can do a lot to educate public and to transfer our knowledge back to the countries where we got educated. I think that's an important part as well, particularly for the countries which are on this lower scale of BDPs and technologies.
Thank you very much, Ivan. That's a wonderful answer. Professor Şahin.
Yeah, I can just repeat what Ivan said. In our company, in our academic institution, we have people from more than 60 countries. Yeah, and they all have their individual stories. Yeah, the individual stories is that either their parents came to Germany, or they managed somehow to come to Germany. Their story is that they are really dedicated. They love what they do. Yeah, and they never gave up. Yeah, and the first aspect is really identify what you like. We are now in a situation where you can really do also learning and what is going on by the internet. Yeah. So the world became smaller, and you can join lectures from MIT, from the best universities, and learn, and really try to get training. Yeah. So practice is important for medical research. Get training in your country. Try to make masters or summer internships in companies. We are getting a lot of applications, and we try really to make it possible to that students could come. And last but not least, stay curious. It is at the end of the day, you have to love what you do. Yeah,
Yes.
and this will give you every day the energy to continue.
Fabulous. Thank you very much indeed. Professor Türeci.
Yes, Ivan and Uğur, they were so eloquent and have basically covered everything. So what I may add is the advice which I give myself every morning when I wake up, and this is about courage and humility as very important driving factors. Courage because, yes, we all know as scientists we need courage to figure out, to go into unknown territory, and in particular if we are in countries where maybe the environment is such that we don't get maximal support, we even need more courage to find our way. And this also pairs very nicely with what is also important for scientists, namely curiosity. Uğur already said this, and if you combine curiosity with courage, it can fly much higher without self-imposed restrictions. And the other part, humility, a quote I like very much is that true humility is staying teachable, irrespective of how much you have already learned. And this is why I have always sought for mentors. And this is also an advice I would give students, also those in low-income countries. Look for mentors, and not only one, look for them in countries you want to go, and stay teachable.
I think Richard froze, so it may be up to me. It may be up to me to bring.
Very impressed. We can see that.
I think this advice, I have never seen Richard speechless, but the advice you have given to early career researchers and students have left them speechless. Professor Đikić, Professor Şahin, Professor Türeci, we cannot thank you enough for your time today. We had a huge number of attendees and so many wonderful questions in the Q&A. I wish we had more time to go through all of them. I want to thank all three of you on behalf of the entire planet, if I may, for giving us faith and belief that we will beat this pandemic. And I learned a lot about mRNA technologies today, and I look forward to all the therapies and cures that's going to come out of utilizing this technology across different disease groups. So thank you very much for joining us today, and I'd like to wrap up the webinar and thank you for joining.